All right, everybody. Thanks again for joining the webinar. We're going to get started. Again, this webinar is going to be recorded, so you can find it on our recorded webinars page of the Novo software website if you'd like to come back and review it. If you could just take another second here, it looks like two of us in the webinar haven't checked in yet in the questions and answers box, but at any time during the webinar, if you have a question, please find the questions and answers box and type your question in there. We'll get back to you as quickly as possible. And for those of you just joining us, again, you can find the questions and answers box. That's where you can ask your questions as the webinar is going today. So I think we have a very exciting webinar scheduled for today. Today we're going to be discussing FCS Express 4 Image Cytometry, and we're also going to be showing some special applications from Dragan Maric. Dragan Maric is a staff scientist at the National Institutes of Health, and later on during the webinar, in maybe about 20 minutes, he's going to be talking about his quantitative in situ mapping of emerging neural cell phenotypes during early rat brain development, using FCS Express Image Cytometry to generate some of these results. But to get started today, I want to show you a little bit about how FCS Express Image Cytometry works, and then we'll progress into what Dragan has done so skillfully with it to get some interesting results. So let me start by telling you a little bit about FCS Express. FCS Express has been developed as a powerful flow cytometry data analysis and presentation software, and we've now been proud to release our image cytometry product. The flow cytometry package has been around for 10 or 15 years now, whereas the image cytometry product came out quite recently. The nice thing is the main principles of flow cytometry and image cytometry overlap, such as taking multi-channel fluorescent data and plotting that data in scatter plots and histograms to look for statistically significant differences and changes. And what we can do is now take your image cytometry data a step further by leveraging the most powerful analytical tools in our flow cytometry package to analyze your multi-parametric data sets, all while relating the results back to the original images for confirmation. So FCS Express Image is going to allow you to spend less time reviewing large, overwhelming data sets and analyzing raw microscopy images by using tools that have been proven to handle data sets with millions of points and allowing you access to the raw images whenever you need. So let's first take a look at the workflow of FCS Express and how you're going to get your imaging data into the software. So essentially what's going to happen is you're going to acquire your images. And these can be any type of images, JPEGs, TIFFs, 16-bit. They can come from any microscopes. They can come from any digital cameras, anywhere that you can actually take an image. And then we're going to bring these through an image segmentation step. Image segmentation software packages such as CellProfiler or ImageJ are used to define cell boundaries, and at this point we can read data from CellProfiler, from the molecular devices Velos instrument, from some benchtop devices from Chemometic, Nexcelom, the LifeTech Tally. We've also announced some collaborations again with LifeTech and with PerkinElmer. And more third-party software compatibility is always in the works, so please check back to our website to see what has been kind of added in the last few months. Keep in mind, though, for this image segmentation step, that CellProfiler and ImageJ are free segmentation software packages, so you don't have to worry about, purchasing one of these, and they work rather well. And once you have your images segmented, what we can do is extract the features, or what CellProfiler or ImageJ can do is extract features such as intensities, areas, things like cell size, cell shape. They can be extracted from the segmentation, and FCS Express can handle as many of these features or extracted parameters as your third-party software can produce. So at that point, we can take this multi-parametric feature measurement data and the images, the raw images, all the segmentation masks, they're all brought into FCS Express at that point for data analysis. And once you're in here with FCS Express or your data analysis, we make it very easy to generate reports, to communicate your results with your colleagues, or to even get you to the steps for publication. So analyzing your data and generating reports in FCS Express is very simple and intuitive. And this is because FCS Express shares many design features with Microsoft PowerPoint. This makes FCS Express very familiar for most users, just because you can create a template or create a layout in FCS Express just like you would in Microsoft PowerPoint. So again, this looks like Microsoft PowerPoint right now, but we are actually within FCS Express. And what I'd like to do is show you around how FCS Express is going to work once you have your image segmentation and your feature extraction done. So to do this, I'm going to come up here to what we call the Applications button, and I'm going to choose to open a new layout. And when we do this, we have a blank page that appears on the screen. And you notice that this is a page, and it looks almost like a page in Microsoft Word. It almost looks like a slide in Microsoft PowerPoint. And it's going to work in very much the same way. We can come up here to our tab and ribbon interface. This is going to act just like Microsoft Office. And just like PowerPoint, we have an Insert tab. Now, in PowerPoint, you're going to be inserting things like shapes, like text boxes, like pictures. But in FCS Express, we want to insert things as well, things like histograms, dot plots, density plots, color dot plots, different ways to visualize your image cytometry data. And to do this type of thing, I'm going to start just by inserting a raw picture, so you get an idea of how this looks and feels. But if I wanted to insert a picture, what I could do is click on the Picture button here, draw a box on the layout where I want that picture to be inserted, and then I can go to my computer here and I can choose where that picture is. And I happen to have a set of images here. And these are the images, the raw images from my image cytometry experiment that I want to show you today. So I'm going to choose this Channel 1 5-minute image, and I'll click Insert. So just like that, we've inserted an image. In this case, we're looking at a protein stain for a particular cell line. And you notice, just like PowerPoint, we can move this box around, we can resize it. So in the past, what you've probably had to do with images like this is kind of look at it, maybe figure out, this cell looks more intense than this cell, maybe this group of cells looks more intense than this group of cells. But once you've done your image segmentation with CellProfiler, or ImageJ, or whatever other type of segmentation software you have, what we can do is bring in an actual interactive image where we can say, let's select this portion of cells and let's look at them represented in a histogram or a dot plot. Or even with a histogram or a dot plot, we can select a region of cells in that plot and then look where those cells came from back in the image. So let's move this raw image over to the side, just so you have an idea of what this looks like. What I want to do next is insert a color dot plot that's associated with the segmentation and the data on this image. So I chose color dot plot. I'm going to draw a box, and I'm going to come to this folder where I have the data stored for this particular image. In this case, you'll notice that the file extension here is cpout. This stands for CellProfiler output. And you notice down here my files have typed their CellProfiler. You'll also notice that with our imaging FCS Express image package, you're going to have access to things like your Velos files, the Life Technologies data folder is in here, Nexalam, ImageJ, all these different formats are available for you. But if we want to choose this file for our dot plot, we'll click Open, and we're going to insert a dot plot, and each of the dots on this plot represents one cell from the segmentation of this particular image. And if I click and hold on the axis of this plot, you notice that we have the ability to choose between any of the different parameters that have been exported by the segmentation here. So if I wanted to choose maybe the area shape area, which is going to be akin to the size of the cell, And maybe down here on the x-axis, let's choose something like the mean intensity of the cytoplasm. So cells that appear in the upper left here are essentially large cells that are dim in the cytoplasmic region, and cells that fall down here in the lower right are essentially small cells that are bright with the cytoplasmic stain. So this is all fine and good, but what we really want to do is be able to kind of relate where these dots are in this image, so it's not so abstract to say, this dot represents this cell. And what we can do is insert what we call a picture plot. I'm going to choose picture plot. I'm going to draw a box, and you'll notice that a picture was inserted in the layout now. I'm going to make this a little bit larger so it's easier to see. Now this picture looks exactly like this picture over here on the left, but the picture plot image on the right is going to be fully interactive with any of the dot plots or histograms or other type of data analysis that you set up within the software. These picture plots are also, if you click on the axis label here, they're going to allow you to choose between the different channels that you've taken with this image as well. So in this particular experiment, we only took 2 images, a cytoplasmic image and the DNA DAPI stain image for this also. As you increase the number of channels, you'll certainly find all of your channels in here. As you'll see in Dragan's data later, he has kind of perfected the technique of working with many, many channels worth of data. But in this case, we're going to leave this cytoplasmic. And I want to show you what we call gating in flow cytometry. Gating in flow cytometry is very equivalent to, in the imaging world, what you refer to as thresholding, where you take you look at a histogram or you look at some sort of two-dimensional plot and you say you want to look at cells that fall above or below a certain threshold. We can do the same thing in FCS Express. What I'm going to first do is insert what we call a gate view, and you'll see that be populated in a minute. But we have something here called the gating tab. And gating is also going to allow us to link up what's going on within this dot plot to what's going on within this image. So I'm going to choose to create a rectangle gate, and I'm going to choose this population here in the upper left. I'm just going to draw this gate, and when I finish drawing that gate, we're prompted to name this, and I'm going to call these large and dim cells because they're high in the area and low in the cytoplasmic mean intensity, and we'll click OK. So the first thing that happens is our gate view is now showing us that this large and dim cells gate appears. You'll also notice that this gate is red and all the dots within this gate are red, or all the cells are red. And also over here on our picture plot, all the cells that fall within this large and dim cells gate are appearing in red. And this means that as I update this gate in FCS Express, that the cells on the image itself are going to be updating in real time as well. So we have this kind of two-dimensional multi-parametric data analysis tool here using gates, which is very much like a threshold. But by using picture plots and doing what we call essentially backgating in flow cytometry, or looking where a cell is by the color of that cell, we can find out the spatial information from the image associated with where that gate lies. And this is all going to be happening for us in real time. Now, this is all good, but what if we wanted to find a particular population of cells within this image and only look at that population maybe within this two-dimensional plot? And let me show you what I mean. Let's say that we only wanted to look at the cells within this polygon gate that I'm creating here. And let's create a polygon gate. We'll say we look at these cells in the middle here, and we'll call these interesting cells, and they'll be blue. So what we've done now is we've created a gate, and they're called interesting cells, and it's the cells that appear here. And what's a little bit difficult to see over here on the dot plot is that there's certain dots here that are appearing as blue. And what I'm going to do is format this plot, and I'm going to change the dot size to a much higher number so it's a lot more apparent to us. And you'll notice that some of these cells now, or these dots, are appearing as blue. And as I move this gate around, those blue cells or those blue dots are adjusting in real time. They say that this is where those dots come from. And what we can do with FCS Express is we can take this gate and we can apply this from any plot to any other plot. And what that means is if I'm only interested in these cells within this gate and I want to look at those cells only within the two-dimensional plot, I can click and hold and drag and drop this entire gate on this plot. And now we're only looking at the cells that appear within this interesting gate. And as I adjust this and I move this around, you'll notice that this gate is updating for us in real time. What we can also do is we can take this gate, maybe this gate from the large and dim cells, and let's ungate this. And if we only want to look at the cells within this large and dim cells gate on the actual image, what I can do is just drag this gate over, drop it onto the image, and now we're only looking at the cutouts for those individual cells that fall within this large and dim cells gate. You notice there's a lot of white background. These are the image masks that are actually being removed in the gating process. And what I can do is if I want to look at this a little bit nicer, I can turn this background to black. And now you can see that we're looking at these on a black background. And as I move around this gate, all this is going to be updating for us in real time. So this is going to be great for finding things like outliers. Maybe we see this one large, this one kind of bright cell here. We know that that cell is coming from this part of the image. Again, we can subclassify these things if we want. Now, another thing we can do in FCS Express is start gating or thresholding through what we call hierarchical gating. And hierarchical gating means that we can look at populations and subpopulations of subpopulations, all sorts of phenotypes of different cells. So let's set that up. And let's say that we only want to look at the interesting cells, the cells that appear blue here, and we want to only look at the cells that are large and dim within that interesting cell population. So to do that, what we would do is drag this large and dim cells gate on top of our interesting cells gate, and you notice that a branch is created. And this branch means that if we apply this large and dim cells gate to the two-dimensional plot, we're only looking at the interesting cells that are large and dim now within this population. We can take this a step further. Maybe we want to view, besides area, shape, area, let's look at the mean intensity of the DNA versus the mean intensity of the cytoplasm. And what we can say here is if we create one further gate, and these are dim DNA, dim cyto, and we'll call these, make these pink. And if we drag this gate again under here, this dim DNA, dim cyto cells, which are these pink cells here, are interesting. They're large and dim, and they're dim for both DNA and for cytoplasmic staining. So by doing this, by setting up multiple hierarchical gatings, you can really have, zone in on whatever type of phenotypes you're interested in looking at within your particular image. And later on, when Dragan presents his data, you'll notice how he has taken advantage of this hierarchical, this Boolean type gating to actually define multiple phenotypes in his experiments. So what I'm going to do here is actually remove some of these gates from the image because I want to show you some of the other features here within FCS Express. One thing that people like to do in the imaging world is they like to look at a histogram for their data, right? We can simply do that in FCS Express by, say, inserting a histogram here. And let's look at the mean intensity of the cytoplasm. And if we wanted to format this histogram to be a little bit smoother, because we don't like looking at all these individual lines, we can certainly smooth this histogram using the format button here to a way that looks nice for our data. Now, just as I gated here, I mentioned that gating is much like thresholding. If we wanted to set up a marker on here, exactly like a threshold, we could create a marker on this histogram, and we could take this a step further by converting this marker to a gate and saying convert and link. We could call these interesting histo cells, and let's make this gate orange. We can see where these orange cells appear, and again, as we update our histogram, you can see how these things are updating on our image as well. So this gating process doesn't just have to work in 2 dimensions, it can work in 1 dimension with your histograms as well. So what we can do is move from this population level as viewed in an image or viewed in a 2-dimensional plot, and we can take this 1 step further by inserting what we call a data grid or a cell gallery. And what I'm going to do is format this cell gallery, and I'm going to show all my gates. And what I want to do is show you the cellular cutouts for a few of these images. And what this cell gallery is going to let us do, it's going to say, let us view all the gates. So an orange block here indicates a cell that comes from this interesting histocells gate. We've chosen to just show the area shape parameter here. So we're showing the multi-parametric data associated with these cells, and we're showing the individual cutouts for these cells. So with this entire experiment, we can do is we can come in here and say, we can find this one interesting cell, we know its area shape, and we know that it's an interesting cell that's large and dim, and that's dim for DNA and dim for cytoplasmic staining. And if we wanted to find all of the individual cells that meet this phenotypic variant here, we can actually apply this gate to the cell gallery and find all of the cells and the individual cutouts and the individual parameters associated with them. Again, if we just want to look at the interesting cells and maybe zone in on just a few of these cells, we can certainly adjust these gates and move these things around. So you can see that we have some statistics being displayed here, just like the raw data being shown within this data grid or cell gallery. At any time, if we want to get full statistics for any plot, we can just right-click on this plot and choose Gate Statistics. And we're going to have all of the statistics appear for this particular data set, like the number of events, the number of cells that fall within each of these gates, the means for anything that you have on the axis, and also all of the percentages of these gates as well. This is going to work for your dot plots. It's also going to work for your histogram. So if I right-clicked on my histogram and chose Insert Statistics, you'll see that we have the high and low bound of this threshold or this marker and everything that falls within it. So you can really get quite complex with your data analysis. What I didn't mention is that we can do custom statistics for any of these statistics that you see. Just like working in Microsoft Excel, if you wanted to do a ratio of, say, your interesting cells to your large and dim cells, you could certainly set that up within FCS Express. There's a lot of features in FCS Express I'm not going to cover today because what we want to do is show you how Dragan Maric has taken advantage of all of these features in FCS Express to start working with his own data. In the future, if you would like to get some more information about FCS Express and more of the features that we have, I suggest logging on to our website at www.denovosoftware.com. And again, we'll come back to this later so you can see the website. But you can go there. We have lots of tutorials and resources, recorded webinars, other ways that you can find more information about what we're doing here. And in just a minute, I'm going to send over this webinar. I'm going to hand it over to Dragan so he can talk about how he's worked with all of these features to use FCS Express. But as I mentioned, we have many resources on our website. And what I want to bring up is that tomorrow we have a webinar with using CellProfiler and FCS Express Image Cytometry joint presentations. I mentioned that we can use CellProfiler for segmentation, and that's what we did in this example. Dragan is using CellProfiler as well. And if you'd like more information about how segmentation is done and how your exports can be created in CellProfiler to bring these into FCS Express, please visit us during this webinar tomorrow. This is going to be put on by the Bird Institute and ourselves. And it should give you a lot of information about working with segmentation for use in FCS Express. So with that all said, I'll pause for a minute. Are there any questions before we switch over to see what Dragan has been up to? If you have any questions, please type them in the question and answers box. And we'll just wait a minute here to see if anybody has any questions. Great, and what I'd like to do, I'd like to introduce and present Dragan Maric. He's a staff scientist at the National Institutes of Health. He's going to be telling us about his quantitative in situ mapping of emerging neural cell phenotypes and what he's been using, CellProfiler and FCS Express, and a little bit or a lot more about the science and the interesting topics that are going on behind this. So Dragan, are you ready to take the presentership here?
Yes, I am.
Great. So let me send this over to you.
Does everybody see it?
Looks like that's coming through great. Thank you.
Okay, so thank you, Sean, for this nice introduction into my domain of trying to use the FCS Express Image Cytometry to get very insightful data from a system biology type of questions that we need to address. And as everybody has seen, Sean has shown what you can do with a single image and all the features that you can plot, extract, and do statistical analysis from that image in FCS Express. I'm going to extend a little bit beyond that and deal with the whole bunch of images that have been generated from slide scanning of large areas on a microscope slide, like an entire embryo or tissues of interest. So, as Sean has mentioned, we're mostly interested in neural cell development and CNS development, beginning with the early stages of embryogenesis all the way through the adult. We obviously, to start with, the most earliest forms where neural tube. Can everybody see the mouse that I'm scrolling around the screen? I hope. We start with the earliest stages of development where the embryos are just basically a neural tube that closes and makes these telencephalic, diencephalic, and so on vesicles, and then study the development from then on in terms of the tissue patterning and birth of different lineages. To get access to those, we take the whole embryos, and what we do is full light spectrum wide field fluorescent microscopy. We're using a scanning microscope, and we use these spectrally compatible fluorochromes ranging from the ultraviolet range of excitation and 440 emissions, such as Alexa Fluor 350, all the way to the near infrared range where we use Alexa Fluor 700, which is the CY7 equivalent. And using immunocompatible reagents like antibodies that detect nestin for immature cells, PCNA for nuclear nuclear marker proliferation, PAC6, which would be a marker of such a transcription factor that drives the cells toward asymmetrical divisions. We use a pan-neuronal marker, in this case tubulin beta 3, and then a progenitor marker that is one of the first commitment stages after neural stem cells start to divide asymmetrically. And using all these 5 channels, we can actually get a nice merged composite image. However, you can see how complicated and how many different organization of cells is within the tissue, and how to actually get access and quantify this is obviously a challenge. So in order to address this question, we actually also have to be aware of the anatomical gradients, what happens when. Like in this case, we have a sagittal section of a E10.5 embryo. We're looking at the PAX6 expression within that embryo. And immediately you can see that there's, like in the region 1, which is a blown up insert here, you can see that green is all proliferating cells, and nestin is blue, but there's no yellow, red, or magenta staining, which means that these cells are proliferating more or less by symmetrical divisions and no differentiation at this point. So by symmetrical divisions, we mean that the daughter cells are the same as the parent cells, and there was no, it's basically self-renewal for short. We can identify just by morphology, the vertical divisions of neural stem cells. There's the other cell types also that are proliferating outside of the brain, which is the extraneural plexus region. Now, in region 2, what we can see immediately is that, which comes from the border between the telencephalon and the diencephalon region, we see just a trickle of the PAX6 upregulation. And what's interesting here, and that's what FCS Express is very useful, is that because we're not just looking at positive and negative, we're also looking at the amount of staining within any individual cell, because that's biologically irrelevant. Just because the cells have a little bit of the PAX6 expression does not mean that they're going to differentiate right away. It is really a threshold, like in panel 3, where we can see a huge upregulation of PAX6, which then kind of over-dominates and switches the cell to truly asymmetrical division. And in panel 4, we can see the end result of that, meaning that the neural stem cells are now dividing into other phenotypes, including the magenta neuroglial progenitors, as well as the first neurons that are being born. And you can see some PAX6 in the neuron, which is tubulin beta 3 positive red cells is here. So this is kind of, at E10.5, you can even count these cells by hand because there's so few cells in any given region. However, a few days later, at E15, and using the same staining protocol, you get a situation like this, and that makes it very tedious and impractical and somewhat subjective of how to, quantify all of these, large numbers of cells. And we can actually see on the right, panel B, that nestin, even though it's all over the telencephalon, we're looking at the insert of a telencephalic region, you can see most of the cells are proliferating. However, most of those are actually dividing by asymmetrical divisions by looking at the PAX6 expression. And what's interesting now is that you get this gradient of neurons being on one side of the telencephalic vesicle. That will be the peel surface, which is also called the differentiating zone, and the neuroglial progenitors, which would be on the CD57 label in the magenta. You can actually see they're all throughout, and they actually begin to kind of express themselves in the ventricular surface and then move on to the by radial migration to the peel surface. There's also tangential migration going from the striatum as well. So, given that all of this tissue patterning is very dynamic and complicated, one of the ways to address this complexity and actually get insight of how this tissue patterning is forming at the beginning of neurogenesis and this is a rat embryo, obviously, we have to kind of initially the old ways were, take individual images and draw little regions of interest and then correlate markers like cell number 7 has nestin, it's PCNA positive, it has PAX6, but it's not a neuron yet, but it's a progenitor. So you can actually see that we can use a matrix, kind of a table, to identify a specific phenotype of interest, and based on that phenotype, do some quantitative analysis. However, doing this manually and by hand, or using some simple, algorithms to identify these cells, just not going to cut it because of the complexity. So, and the sheer number of cells that need to be quantified. So the way to approach this in a more practical sense is to actually go in, and obviously we can analyze it like Sean has shown you in his one image example. A single image at a time is relatively simple to analyze, but when we deal with huge areas of tissue that we need to analyze, we need to actually segment that or slice it up into manageable tiles. And what we do is, this is a shareware that you can go and on Google and find it and download it. We use a total image slicer to take these larger areas of interest, like this is the insert of the telencephalon, which we'll focus on for this example. We import all of the channels, or into the image, a total image slicer, and the next step is to select the destination folder where all of these sliced tiles are going to be stored. We can set up the sizes of the slices that we want to break the big image apart, and in this case we're just using 1,000 by 1,000, although you can use whatever. You don't want to have too large of slices, obviously, because they'll be a little bit harder to handle in CellProfiler, but it's 1,000 by 1,000 seems to work very well for us. Obviously, when the large image has been sliced, it's going to have rows and columns, which will be then read through CellProfiler, which is the next step. You don't want to. You can compress, but there's no really need for any compression, and you can leave defaults as they are, and you just convert all of these large images into slices. That's very straightforward. So once you have all the big image sliced into manageable tiles, the next step is to import these tiles into CellProfiler. As Sean has already mentioned, CellProfiler is you can go to their website. It's very well, packed with information, and you can always ask questions about how to best apply the segmentation to identify individual cells, as well as cell nuclei and cell bodies, and measure all the extraction features that you would like, or intensity, shapes, area, whatever you need. It's a very versatile program to extract features from the images. In this case, obviously, once we import all of these images, we have to identify primary objects, and typically the first primary object is the nuclear. You can use either a nuclear mask, or in this case I use the cell mask, and then set up which cell mask is just basically a composite of all the different stains that we used in imaging that identifies all the proteins or all the cellular compartments of interest. And the nuclei in this case have been used to be identified by cell size or nuclear size. And you can see in the panel on the right the color-coded images, and these color codes are just conveniently, they don't mean different fluorescences, they just mean different segmentation, that the segmentation has identified all of the individual compartments, all of the individual nuclei, or all the individual cells in this image. We then go and identify, and again, you're going to have to kind of go ahead and see which feature will be the best, or which algorithm will be the best to extract the best features and the nuclei, and you can actually see that by looking at the sample image, by using a test run on the CellProfiler. After you identify the primary objects, obviously you want to identify the secondary objects, and that is typically the input objects will be nuclei, and then you can do different ways to, you can expand from the nuclei to identify individual cells and use the bright field image. In this case, you can actually see that the individual cells can be kind of demarcated by the phase contrast image as individual circles, and that can be used as a nice way to identify individual cells. So you don't even need cell mask in that case. Sometimes cell masks are not very reliable when you have very densely packed cells that overlap each other, but the bright field seems to be working well in this case. The tertiary objects obviously can be a difference between the nuclei and the cell areas, and you can actually see on the color-coded panel is that the first panel is the nuclear, then you have the cells which are bigger than the nuclei. You can see those areas are kind of larger, and then the difference would be the rings, which would be the cytoplasmic compartment. And so what we are doing here is we're basically identifying all of the cells based on the nuclear compartment and identifying their cell bodies, and then the difference would be sort of like a cytoplasmic compartment. So once that has been kind of processed, you can then measure, add your parameters of interest. In this case, we have our 5 channels: nestin, PCNA, PAC6, tubulin beta 3. Obviously, the objects to measure will be the cells in the nuclei as well as the cytoplasm. So all of the intensities of individual fluorescent stains would be then measured within all of these compartments. These will be very useful if you're looking at trafficking of molecules from one compartment to the other. Nuclear translocation, comes to mind, or many other different. Or if you want to look at if the compartmentalization is really nuclear versus membrane, and so on and so forth. So after we kind of went through all these, things that we want to measure. Oops, what happened here? The first thing is that, we have to analyze all of these images and extract all of the features. And from there, we can then import the data into FCS Express for multiparametric image analysis and data presentation. So the first thing that we do is, these are, on the top panel is our original images in 5 channels. And just like in flow cytometry and in any field-based microscopy, you really have to identify your positive and the negative population. And the beauty of this FCS Express is that you can use regions of interest to identify areas where you would have negative cells. So there's obviously cells throughout this tissue, but if you draw a region where there's no specific staining for a given marker of interest, you can actually then gate those negative cells and display a histogram of the mean intensity of those negative cells. Why is this important? Because once we have the mean intensity and a histogrammatic representation of the range of autofluorescence of negative cells, we can sort of create a marker region of interest where we identified the thresholds, like Strong has mentioned in his presentation, where we have a boundary where the brightest autofluorescent cells kind of end, and then above that would be then the positive cells. And in order to verify, we then show the ungated histogram in the second, so these are the third row. And ungated histogram will actually show you the negative area and the positive area, but because we already have identified the threshold for the negative, we just kind of extrapolate that and then extend the positive region away from there to the maximum fluorescent population. And once we have identified both the negative and the positive cells, we can then recreate an image plot where the cell mask as a background, and you can see the cell mask is sort of like a grayed out area where all of the cells have been shown. And then a color-coded, because we color-coded the positive cells in green, you can actually see that you can reproduce the original image on top with the masked image on the bottom, and you can see it's exactly the same image, but now we have quantitative assessment. And in this case, we can even have a quantitative number, like in this case we have 40,000 cells in the total image, with 13,000 cells representing 32% of the total being PCNA negative, and the remaining population, in this case 27,840, which represents about 68% PCNA positive. If we now use the same logic for the rest of the channels, obviously PAC6 sort of like has the same area of negative cells as the PCNA, and by drawing, again, a negative and a positive region, we can reproduce basically the same as the original image. Nestin, of course, that's a third row image. You can see that that's going to be difficult to get negative cells inside the cortical tissues and the striatal tissue because nestin is all over the place, but you can go outside, and you can see here there's a lot of cells outside of the cortex which are kind of gray, so you can draw your small region of interest nestin, for nestin negative cells. Obviously, we only have a very few cells here. And you can see this is bin data 1 and 2, so you can actually, it's so sensitive that you can identify 1 cell and 2 cell kind of bins, and obviously draw your negative cells around. And it's even good enough if you have like 50 or to 100 cells to kind of get a feeling of where the negative cells are. And then the positive cells are obviously the rest of them. And again, the idea here is to reproduce the original images in the from the CellProfiler segmentation, and reproduce the masked image with a specific population of interest in the with the FCS Express. Once we have done that, now this itself presents a lot of information. You have PCNA positive cells, you have PAX6 positive cells, you have nestin, and all of this is already quantitative. And if we were only interested in individual single phenotypes of cells, this may be enough for many applications. However, you can extend this way beyond this, and now you can really have some fun with the data analysis, because just with flow cytometry, once you have identified the positive and the negative gate, you can use Boolean logic in order to identify multiparametric analysis. So in this case, if we were to combine PCNA positive cells, PCNA positive and negative with a PAX6 parameter and just plot a bivariate plot, and also we can do this with nestin and CD57 and CD57 and tubulin beta 3, we get to this panel of data analysis, and that is a bivariate plot where on the x-axis, in this case, PAX6, and plotted with the PCNA. Notice I use the log scale because that allows me to plot very low events and with very high events all in the same plot, as well as also getting some sort of a Gaussian distribution, which is sort of like a population kind of base that you would see on a flow cytometric analysis on a histogram with a log scale. And what you can see there on the second, in the middle row, you can see the panel of PAX6 distribution with PCNA, and based on our thresholds of marker regions, we can then use the same thresholds on the quadrant regions and identify the double negative and the single positive PCNA cells, the double positive cells that are also dividing asymmetrically in the orange, and then we have these PCNA negative PAX6 positive cells with pseudo-colored in yellow in the lower right-hand quadrant. With this, we actually have all the quantitative analysis, much like the flow cytometry data, with the total number of events and characterized on the double positive, double negative, and single positive cells, and well as percentages of the total cells out of this population. And now, what's very interesting is that you take this, and something that conventional flow cytometry can never do, is we actually have spatial information from this bivariate plot, because not only do we have accountability of all the cells inside that tissue, we can then pseudo-color the patterns of cells that are actually in the back in the cell mask image plot, and you can actually see and reproduce not only where the cells are, but where their specific phenotypes are. So, for example, if we're looking at PAX6 PCNA positive cells, here they're color-coded in orange, you can see that they're basically patterned within the ventricular, subventricular zone of the telencephalon, and they're absent in the striatum and in the cortical plate, subplate future regions of the peel surface of the telencephalon, which is extremely interesting. And then when you gate on these PAX6 positive PCNA negative cells, you can actually see that they're actually patterning in a future rostral migratory stream, which is actually the migratory neurons that are actually going away from the subventricular zone and entering into the rostral migratory stream, and then to populate the olfactory bulbs. Using the same logic, we can look at in the other parameters in bivariate plots and recreate different patterns of phenotypes based on these bivariate plots, which is a very, very elegant way to identify like a multiplex analysis with multiparametric identification codes for cells that are, sort of like very unique in phenotype. And why is this important? Because just because PCNA is a marker of proliferation does not necessarily mean that it's unique to stem cells. Other cells can proliferate inside the brain as well, and it's definitely, or progenitor cells as well, and it's definitely nice to combine these markers to identify specific phenotypes of interest and the state of their differentiation or proliferation. Now, one step further, and this is the ultimate, and is you can now combine any marker with any marker and create a matrix plot. So everything with everything. In this case, in the upper right-hand panel, if we really want to identify neural stem cells that are dividing asymmetrically, they have nestin and PCNA, but so other phenotypes can have nestin and PCNA, including the progenitors. What they don't have is the PAX6, which is driving them for asymmetrical divisions. They don't have a neuroglial progenitor marker, CD57. They don't have the tubulin beta 3 marker, which is a marker of neurons. So once you add this Boolean logic, positive for nestin, positive for PCNA, and negative for everything else, you can pseudo-color it in green, and you can immediately see, very few kind of stem-like cells. Obviously, this is not perfect because, we only using 5 markers. We can actually use now up to 10 to identify the endothelial cells, which are also nestin and PCNA positive cells, and exclude them from this gate to actually make a more precise identification of stem cells. However, when we look at early neuroglial progenitors, those are much more unique in that they're nestin positive, PCNA positive, and PAX6 positive, but they're still not committed to CD57. In this case, we put them in yellow, and you can see them very nicely patterned within the ventricular-subventricular zone in the telencephalon. Late neuroglial progenitors are, the only difference is that they do now express CD57 in addition to PAX6, PCNA, and nestin. So you can see we have 3 populations that are nestin positive, and all of them can be PCNA positive, but the differentiation matrix here says stem cells are negative for PAX6 and CD57. Early neuroglial progenitors are positive for PAX6 but negative for 57, and late neuroglial progenitors are positive for both. But they're still negative for tubulin beta 3, meaning they haven't differentiated into neurons. And in this case, we put them in magenta, and you can see them in a quite a different, unique pattern here that is sort of intermixed with the neurons and the early progenitors. So, like Sean has mentioned in his presentation, is that you can do this hierarchical gating, much like in flow cytometry, where you can actually put, first of all, you can do the entire telencephalon, in this case, analyze the entire region, but what's most even insightful is you can do subregions of interest, such as, in this case, we do the ventral telencephalic region versus the dorsal telencephalic region, and then we have WTC, which is the whole telencephalic region, and we can then gate or put all of these populations that we identified with the matrix under each of these regions and get quantitative numbers for the absolute numbers of cells, the percent gated cells within those regions, as well as percent of total cells of the total area of interest. And using these numbers, you can then plot them as the bar graphs and getting percent of total and the total number, and very nice publishable plots where you can extract from the FCS Express. And as Sean has shown you, you can take one of these regions and move them around the telencephalic, and they will update automatically, which is also very nice. So you only have to identify one or two of these regions and kind of move them around and get the data updated if you want to move to a specific area of interest, or you can just leave the ungated area and look at the total numbers as well. So that will be sort of like one example. Obviously, I mentioned that this is a little bit oversimplified example, but it illustrates the principle of how to use FCS Express to get very nice spatial information, as well as quantitative assessment of tissue patterning from different areas of interest. So with that, I will end this portion of my presentation. Sean, back to you.
Hi, Dragan. Thank you very much. What I'd like to do now is open up the floor for any questions before we kind of summarize what we went over today. Are there any questions while Dragan's presentation is fresh in your mind that you may have for him? If you have any questions, please locate the questions box in the GoToMeeting panel. You can type your question there. So while questions are coming through, I'll take the presentation back here just to summarize. So what you saw today in Dragan's presentation and in my kind of basic intro is that FCS Express is going to leverage some of the most powerful and proven tools of flow cytometry analysis, and there's lots of new features to help improve your image cytometry analysis as well. FCS Express Image is also going to allow you to move from that population-based analysis to single-cell analysis at the click of a button. Besides population-based analysis, you saw that Dragan can move around his gates for the different portions of the image that he's looking at, and you can move to see what's going on between your different populations very easily. All the image handling features allow you to act fully interactively between all the data plots and all of your images and the cell galleries and the bar plots and all the gates and data that you've seen within the software also. We also offer a free reader and online collaboration tools for report sharing, so we make it very easy to actually collaborate with your colleagues and when it comes time for publishing, if you need to share your analysis with other people, we make that all very easy. The Microsoft PowerPoint feel makes the interface of the software very easy to use and understand really for any level of user. And in the end, we provide many resources for our customers like tutorials, sample data, and online forum. And we also like to say that we have a very great customer support system so you can get up and running with the software in no time. If you have any questions about the software, if you're trying it out, if you're trying out the tutorials and the sample data and you run into any questions, you can always email us at support@denovosoftware.com. And more importantly, if you'd like to try out FCS Express Image or Flow or the Plus version, which is FCS Express Flow and Image, you can get this from our website at www.denovosoftware.com. We are offering a free 30-day trial, which is fully interactive. It comes with all the features. You can see this. This is in our website here at Download FCS Express Demo. You can come here and get the demonstration version for you, and again, you can go in there and try with some of our own sample data if you want. And I also mentioned that we have lots of resources for our customers. One of them is webinars, and as you saw today, we had the FCS Express Image Cytometry special applications. But if you're interested in how you can export your data from CellProfiler, or you're interested in learning more about segmentation, you can join us tomorrow for the Using CellProfiler webinar. And we actually have a whole series of webinars going on this week. On Wednesday, we're going to be working with Nexcelom image-based cytometry, so if you're more interested in the benchtop side of things and you have a Nexcelom Vision CBA, you can join us there. We also have a nice, another special applications example from Erlu, who is at La Jolla Biotech Institute. He's going to show us how he's using FCS Express Image Cytometry for Raman scattering and different things like that. And on Friday, we're also going to be going over using ImageJ and FCS Express Image Cytometry. So as I mentioned, CellProfiler is not the only way that you can get your data into FCS Express, or it's not the only free way. You can also use ImageJ. It's something that we released in the last few months, so it may be something that you're interested in. So again, visit our website to get the free download if you'd like. On our main page, you can find the registration for all these webinars right up front if you'd like. We also have some nice videos. So with all that being said and kind of summarizing, again, I'd like to open up the floor for questions for either myself, the De Novo software team, or for Dragan Maric. You can enter your questions in the questions box in the GoToMeeting pane. So if you could find a minute to find that box if you'd like to ask any questions to myself or Dragan, we're both still on the line. And if no questions come in, if after the fact you'd like to either contact us or contact Dragan, please contact us at support@denovosoftware.com. We'd be happy to answer your questions there, to get you in touch with Dragan if you have more questions about his use of the software or the scientific backgrounds of anything. So at this point, it looks like there's no questions on the line. And thanks again, everybody, for showing up. We really appreciate you coming by the webinar today. Again, I hope you can join us for some of the other webinars we're giving this week. I'm very much looking forward to working with the Bird Institute tomorrow to show everybody how you can use CellProfiler and FCS Express. So we hope to see you there as well. And again, it looks like there's no more questions, so we're going to be ending the webinar today. Thanks again, Dragan, for your presentation. It was very insightful. It was great to see what you're doing in FCS Express and how you're putting the software to great use.
Thanks, Sean, for letting me do it.
All right, everybody. Have a great day, and we hope to catch you at the webinars later this week. Take care, and bye-bye.